Looks like we're up.
Great.
Hello, everybody. I'm Karl Haro von Mogel, and with me is,
Hi, I'm Anastasia Bodnar.
and we are coming to you both from Wisconsin. And where are you at right now, Anastasia?
I'm in not so sunny Virginia Beach.
Ah, so we're here to talk about the GMO Corn Experiment to do a live Q&A. We've gotten a lot of questions from people since we started this fundraising campaign for this project. A lot of interest from people, and so we're hoping to clarify some things. And if anybody has any questions, feel free to put it in the Google Plus Hangout chat on our page there or on the YouTube channel, and we will try to answer them as best as we can. So, all right. So let's. So, how excited are you to get this project going, Anastasia?
Well, you're the one who's been doing most of the work. The corn is not in my basement. Um,
Yes.
but I am really excited to start getting the data come in. I mean, this is a... Sorry, that's my daughter Rose in the background, of course.
She's
Um,
super excited as well.
she is very excited. This is something that we've been talking about for, gosh, it seems like at least a year,
Hmm.
if not more. And we really have an opportunity to take one of these like great internet, myths or questions and, really
Hmm.
get some data for it, which is, I mean, come on, everybody loves data, right?
I love data. Hands up if you like data.
Yeah, I love data.
So, and not just on Star Trek. So, yeah, so you said the word myth, but I like to say the word claim because we're testing the claim that animals will avoid GMO corn and prefer non-GMO corn if given the choice. And we see this everywhere from farm animals to squirrels and chipmunks and such. And so that claim floats around. People talk about it, but nobody's actually done a study to test it. And so for a lot of people, it seems kind of doubtful, like, because there's 94% of the corn, the field corn grown out there is genetically engineered. And if the animals were avoiding it, then that really wouldn't square up with how many animals are actually eating it. And there are farmers that have certainly not claimed that their rat and their rodent problems have gone away since they started growing GMOs, or their deer have decided to steer clear of their farm. That's certainly not happening. But you do see those pictures out there of a couple ears of corn next to each other, and nobody's actually really tested it scientifically. So I actually went tracing through my emails. So, Anastasia, you mentioned we've been thinking about this for a year. I found an email from 3 years ago
Oh
where I
my God.
was like, this is what I want to do. I want to do this experiment. And
Wow.
we tossed around the ideas. I know Kevin Folta and Kevin was not able to make it for this hangout. Kevin Folta and I have been talking about this for a while, but it was a year ago in August when I remember I sent the both of you an email saying, let's do this thing. Let's get the corn and let's do the experiment. And if we can't get the corn, then that's a story in and of itself.
Right, it
But,
is. So
yeah.
can you tell us about that? I mean, I know some of the story, but how hard was it to get Big Bad Monsanto to give you the corn and let us do an experiment on it?
Well, so I had to give about a pint of blood
Oh
to be able to
no.
get it. No, actually, so yeah, this is one thing that we wanted to actually take a look at and investigate. And there was a Scientific American article in 2009 where they were saying that there were some researchers who said that they weren't able to do the studies they needed to do on genetically engineered crops. And they would say it's because Monsanto and the other biotech companies have these patent rights and they're not able to get the actual crops to study them.
Yeah.
And what came out of that was, first of all, a realization that many of the scientists didn't realize that they actually could get them. Many of them hadn't necessarily tried, but also it wasn't too easy and straightforward for them to do so. And so some of the companies, they came forward and they drafted more detailed and open agreements, particularly with universities and USDA researchers. So when we inquired from Monsanto, we decided we'd approach them because they're one of the big companies everybody's talking about, and everybody talks about whether or not Monsanto will let you get a hold of their corn to do an experiment. So we thought, let's approach them. We approached them, and I was talking with them on the phone, and I mentioned I was at UW-Madison. But I wasn't doing this through UW-Madison, doing this through Biology Fortified. But at the time, I was working at the university, and they said over the phone, "Oh yeah, you're at UW-Madison, no problem. Tell us what you need, whatever the experiment is."
That's
and I'm like,
great.
"Oh, what?" And I'm like, "Oh, wait, this isn't through UW-Madison." See, they have a standing agreement with the University of Wisconsin. So the public researchers can just call them up and go, here's what I'm going to do. Here's what I need. And they will send it to them. They've got sort of a blanket agreement with many of these universities, and University of Florida and other universities. But, so since we weren't doing this through the university, doing this through Biology Fortified, I'm like, well, we don't have a standing agreement with you, so we need to go through that process. So I gave them a description of the experiment of what we wanted to do and what we needed and what the requirements would be. We need 2 different varieties of corn, one of them genetically engineered, one of them not. I wanted to, because we don't know what trait in the genetically engineered corn might be responsible for this supposed squirrel avoidance. So I thought, let's get all the traits we can. So I asked for something that was multi-stacked, that would have many different Bt traits. Bt is an insect resistance trait, and then several different herbicide tolerance traits. And those are the ones that you're usually going to find in corn today. So then they came back to me with a material transfer agreement. First, for a little sample, I did a little experiment with just some loose kernels in my backyard. And so they gave us a material transfer agreement for that. And then they proceeded to plant and grow the corn in Hawaii according to our specifications. And we went through a couple of rounds with talking about this transfer agreement and making sure that it had the details in there that would satisfy them and would also satisfy us. And there was a little bit of fun and frustration involved, because they're used to dealing with very straightforward experiments. They provide a material, the scientist does the study, and then when they're done with the study, they submit the study into a peer-reviewed journal. Their normal boilerplate material transfer agreements involve simply just giving them the results before you submit it to publication. They don't have any veto power over your ability to publish at all. You're not granting that, but you are granting them access to your results.
They just want a heads up, I think, is what it sounds like.
Yeah. And a lot of people think that might be because, what if you find a problem, then they want to be able to respond to it. I actually think it's something a little less sinister than that. They're working on things that are patented. And if you come up with some new discovery based on their patented thing, they want to know what you've discovered with their materials. So there could be some sort of discussion about, okay, now who owns this new discovery based on it? That's what I think a lot of that has to do with.
You could also, any of these studies could have a result that would affect their business. Even if it's something good, it could affect their business. Maybe they need to roll out more seed production, so it runs the range from that negative findings for them or positive findings for them. I think it makes sense that they'd want to know in advance. But the question that I have is, I wonder how much it cost for them to produce the corn and ship it from Hawaii to Wisconsin. I appreciate that, that they went to that expense, especially because they don't know what our results are going to be.
Yeah. And during this whole process, I explained to them, this is going to be a very open and public experiment. And so when it came to some of those results, their normal boilerplate transfer agreement would say, you need to tell us 60 days in advance before you post and, you know, publish anything. Well, I'm like, well, what about a couple of pictures on social media? Do we need to give you 60 days advance notice before I tweet a picture of this? And they're like, oh yeah. So, you know, I sent back my comments, and they changed the material transfer agreement in order to make both their patent lawyers happy with their stuff, and to make us happy with being able to do the experiment in the manner that that we needed to. And so I have read about some researchers who have inquired about getting genetically engineered materials from some of these companies, and they get that normal boilerplate back, and they go, oh, I don't like this, and they go, forget about it. And so that's one of the things I hope to make people aware of, is that's their normal starting point. And then you go, well, this isn't going to work for me because of this and that. Can we change it in this way?
Right? I mean, it's just about having a conversation.
Yeah. And I think we got a pretty good one. So people, you were going to be able to put things out through social media that's explicitly in there. And we made sure some of the language was very clear, that that, you know, they're not going to know who we're sending the corn to. So everybody's privacy is maintained, and just a whole bunch of little things like that. And so we're going to actually post the material transfer agreement on our website for everybody to see.
Oh yeah, so that's a good point that we should make clear. We're going to send them our results, but it will be de-identified results.
Yeah.
So people's names and addresses and all that information will be stripped out.
Yeah, and we're going to post all the results online. We're going to figure out a way to display these pictures somehow and the other data that we're going to generate, and so everybody can see what all the data is, in addition to the conclusions that we draw from it. So it's going to be a huge public thing.
So I think that's a really good time to talk about what we're going to do with the data.
Yes. So I think you can say something about that.
Yeah. So we actually got a really generous offer in the information about in the experiment.com. Right. So we got some great information. We got an offer from some students, a student group at University of Wisconsin. It's called
I think it's Minnesota, wasn't it?
Oh yeah, sorry, Minnesota. Yeah. Statistics in the Community, I think is what it was called, something like that. And I feel terrible because I'm looking through my email really quick, but I have so much going on that I'm having a hard time finding the exact name of it. But anyway, so it's a third-party development with, you know, and they're going to be doing our data analysis. And so, yeah, it's Statistics in the Community. Awesome. I did remember the right name. And so you can Google them, University of Minnesota Statistics in the Community. And their whole mission is to do statistical analysis for nonprofit organizations. And we're a nonprofit. They mostly focus on local groups, but they're making an exception for us, which is very nice of them. And so
I'm local enough. I'm just a 4-hour drive away.
Right, you can drive out and say hello. But what's nice about that is that, you know, the people who are collecting the data, that's blinded. So all of our citizen scientists, they won't know if it's a GMO ear or a non-GMO ear. However, if Karl and I did the data analysis, you know, we would have, sorry, my daughter's asking for something on the iPad. You got stuck, didn't you? Okay, there you go. Okay. So, you know, if we did the data analysis and we knew which ears were which, you know, if that was in our big data file, then it wouldn't be double blinded. And so, you know, of course we would, you know, do it as in an unbiased way as much as possible. But, you know, we want to be as rigorous as we can. And so thanks to Statistics in the Community, now we're going to have a double blinded design where the people who are doing this statistical analysis, they'll know it's A and B, but they won't know if A is GMO or non-GMO or B is GMO or non-GMO. So I think that's a huge benefit that they're bringing to us. And we also appreciate their statistical expertise. And what's really another great benefit of this is that we'll be working with graduate students in statistics. So, you know, part of our, a huge part of the benefits of what we're doing here is being able to interact with a lot of students. And this is just another layer of bringing on some graduate students to work with us.
Yeah, and you know, we wanted to find some graduate students in statistics to do this, and you know, once this thing got launched, I was thinking, okay, now we get the funding, I have to go find them, and turns out some of those students then came to us and said, "Hey, can we analyze your data?"
Pretty awesome.
Yeah. So Anastasia was talking about blinding. So let me do a little show and tell. I'm going to show what. Don't pass out yet, Frank. We're not done. So I'm going to do a little show and tell to show what the experiment kits are going to contain that people are going to get. You know, we had the video, we had some pictures and all that, but I'm going to show what everybody's got. So what everybody's going to get in the mail who participates in this experiment is they're going to get this fancy dancy feeding stand. So I've been making these in my garage. Got a nice piece of 1x4 and pre-drilled with holes that will very neatly fit a couple of nails. And those nails, when you put them in, will then be able to hold up the ears of corn. Now, the ears of corn are going to come in individual bags. In every kit, there are going to be 2 pairs of ears labeled as Experiment 1 and Experiment 2. And you'll see a couple of colored dots on these bags. These won't be actually on the corn when you get them, but those colored dots actually refer to whether they are GMO or not. And I won't say which is which, but that's just what we're doing to keep track of them as we put the kits together, and then those will be removed. So then we can make sure that we know which is which when we're putting the ears into all the kits. But then when everybody gets them, all they're going to see is Experiment 1, and there's going to be a couple of ears. So then you'll choose which ear you put on the left or the right, and then take a picture and upload that to a nice, easy-to-use web portal that we're setting up. And then you'll be able to record your data in there, take a picture a day later when it's done, and Anastasia will be able to talk a little bit more about what kind of data we're going to gather. But it'll be real simple and easy to do. And then when that experiment's done, then you can take the ears for the second experiment and put it in there. Now, when you're actually putting them on the feeding stand, in each bag, we have a barcoded tag. And that little barcoded tag you can stick on the front of the feeding stand, just like I showed in the video on experiment.com. So this will allow everybody to know, we'll be able to track the ears from beginning to end. And when it comes to analyzing the data, we'll be able to tell the statisticians who are looking at this, these are all the ears, this is their identity, this, genotype A and genotype B, and then they'll be able to tell us, is there a difference? Is one of them getting eaten more than the other? Is one of them getting ignored and while the other one's being eaten? Or are they both being eaten equally, whether completely consumed, partially consumed, or not even touched? So yeah, that's how that's going to work, and that's going to keep it so that every individual person doing the experiment won't be able to accidentally or purposefully change the results because they won't know which one is which. And then the people doing the analysis won't be able to bias how they do the analysis because they're not going to know which one is which. And then they'll tell us what the results are, and we'll have to accept it one way or the other, because that's science.
Exactly.
Because science.
Yep. So you mentioned what kind of data we're collecting, and I'd be happy to talk about that a little bit. So the basic data, like the data that we want everyone to send in, will be the pictures, and we'll work with statistics in the community once we're assigned statisticians, which should be within the next week or so. We'll work on a way to use an unbiased analysis to to look at those pictures. So we'll ask everyone to get down on the ground with their iPhone or camera or whatever they have available and take a picture of the stand before. And so that should show, you know, the board with 2 ears and the barcodes. Take a picture pretty close before, and then they'll wait 24 hours. And then take another picture from the same place showing the barcodes and the ears of corn after 24 hours. And that's the basic deal. We'll probably use some sort of image analysis software to see what percentage of the ears were consumed during that 24-hour period. But that's not all. I think that we're going to have people who submit additional data. A lot of people are doing this for a science experiment, for science fairs at their school, which is great. We're so excited to be part of that and, you know, help kids all over the country, all over the United States, work on their science experiment. Yeah. So we're going to find a way to help to collect that data as well. Oh, sorry, Rose wants to be picked up. Come here, baby. Okay. Can you sit and watch Karl?
Is Rose going to participate as well?
Maybe. Basically, we're going to, you know, make a way for everyone to submit additional data in addition to that first picture and second picture after 24 hours. No, honey, you can't play with the mouth.
Yeah, we're going to be adding in a couple other things, and the main important data will be the images, but there will be other things that people can do, such as after 24 hours, count the number of kernels left. You could, if a child is doing a science fair project around this, they could also measure. They could take additional pictures like every hour during the experiment, and different ways.
They could take the weight before and after of each ear, so they could, you know, what the weight difference is, count the kernels, take additional pictures. So there will be a lot of different ways for them to, you know, collect additional data and use it for their science fair project, and then also submit it to us. And we'll have some standardized fields so that if enough people submit that kind of data, hint, hint, we're encouraging everyone to collect additional data.
Yeah.
Then if we have enough, and the statisticians will tell us, then we'll be able to do the statistical analysis on that too.
Yeah, and I'm really interested to see what kinds of other data and ways of collecting it that some of the other participants are going to come up with. We're going to give some suggestions, but you know, there are going to be a bunch of things that aren't, you know, required that people may want to do.
Yep.
Yeah. So I think we got.
Do you have any questions from Twitter or anything like that? Are you?
Well, I don't see anything directed at our Twitter account or mine just yet. I do see a comment on the Google Plus Hangout page. Jefferson points out he's suggesting that wood screws would be safer than nails. And yeah, that's actually a pretty good suggestion. One of the issues, you know, we were thinking about even using wooden dowels at one point, but I found that it was extremely hard to get the ears on and off the wooden dowels. For the nails, they're not as sharp as the wood screws are, and we are going to make sure that that adults would be the ones putting the ears on the feeding stand, and I'll give very clear directions on how to do it. You know, even though you are talking about putting something on a nail, your hand can be very far from anything that could possibly be injurious. But with a screw, while you could slowly twist it in there, at the same time they are sharper, and there's no perfect solution to this conundrum. And even for the wooden dowels, I was thinking about using those sticks that are used for caramel apples, because those have a pointed tip as well. So I got some caramel apples, ate the apples, and then tried out, tested it out with the sticks. But like I said, it was so hard to get the ears on and off. I thought those could even just splinter and injure somebody. But the nails are actually very, very smooth, and they slide onto the cob. The cob slides onto the nail very easily without very much force. So yeah, there's tons of considerations involved. I mean, we also have the issue of the safety to the animals to consider. Did you want to say something about that, Anastasia, or shall I?
Well, I guess I can talk a little bit about. We definitely don't want people to approach the animals.
Yeah.
So it depends greatly on what type of environment you live in, whether you're urban, rural, suburban, what part of the country you're in. But who knows? Maybe your corn will attract some bears. I mean, we don't know what's going to happen out there in everyone's backyards. We definitely don't want people to approach the animals. We want them to be careful, stay inside, and make sure that you look around and that there aren't any animals around when you go and approach the corn if you're taking a picture of it.
Yeah. Yeah, we don't want anybody to be handling the animals. And we actually went through a process to make sure that this experiment would be okay for the animals. Whenever you're doing animal research in the United States and in other countries, you have to make sure that you're actually not going to do anything that would harm the animals. And so the way that researchers do that is they talk to the institutional review boards at their universities. And so because Kevin Folta is at the University of Florida, that's our anchor to a university there. So we went through that process at the University of Florida, and we got a document from them telling us that, yes, this experiment that you guys are doing is okay. You don't need to go through any really strenuous process to make sure that to ensure the animal safety, like a lot of other studies do. Go ahead.
That was a pretty easy process for us, is that all of the biotech traits, all the GMO traits that are in these ears of corn have all been thoroughly examined by the FDA and have been tested for safety. Well, so the companies provided information to the FDA demonstrating that it's safe for food and feed purposes, and the FDA analyzed the evidence and agreed that it was safe for food and feed. This is not experimental corn. It's very similar to the corn farmers are growing all over the country and all over the world.
Yeah, these, we selected these varieties and this corn to be able to use corn that the farmers are growing right now. So none of these are like experimental traits or something like that, haven't been approved. There'd be no way that we would get those kinds of traits from any of these companies anyways before they've gone through the approval process. Yeah, let's mail out your unapproved corn to 1,000 volunteers around the country, and we won't tell you who they are. Yeah, nobody's going to do that, right? So yeah, these are all.
Appreciated that either.
What's that?
I don't think the government would have appreciated that either.
No, because again, if you're going to move experimental materials, you have to get approval for all of its movement, and usually it's in locked metal boxes. So that would have added a tremendous cost and hassle to the experiment. So yeah, this is essentially a wildlife observation study with a little bit of human involvement of leaving out some corn. For other kinds of experiments where you're going to be, having confined animals and all sorts of things, euthanizing them, also that kind of animal testing stuff that requires a lot more review. And this is a very simple experiment, and the corn has all been grown for human and animal consumption. Don't eat the corn, by the way. This isn't for you to eat. This is for people to leave out for the squirrels and such.
If you wanted to, you could, but it wouldn't taste very good. I mean, it's field corn. It's not. It's grown for processing and for feed. It's not grown for food purposes.
I have to confess though, I did nibble on a couple of loose kernels, and it just tastes like field corn, like you would use to make cornmeal. And I can't quite find a difference in flavor between these two, so that's good. But we might end up adding a little flavor test to this just to make sure that if we find a difference, it's not because one just happens to taste better or something. We'll see.
That's a great experiment.
Yeah. Yeah, so we got another question from Bill. He said, can you address the idea of transgenic and isolines and how they will be used here? Yeah, so that's a great question.
Uh-oh. Lost Karl for a second.
Okay, I'm back. Can you hear me?
Great.
All right.
Yes.
Yeah, it's a great question if I don't accidentally step on my microphone cord and knock it out. So yeah, if you're going to, in science, find a difference between two things and do take that difference and be able to say that this is due to some trait that you're interested in, you have to control your variables. So when it comes to doing an experiment as this, like you've seen, we showed those pictures of ears of corn that people left out for squirrels. Though usually their description was, I just got an ear of corn from this farmer and I got these ears that were sitting out in my barn. And look, they liked one more than the other. Well, there are all kinds of different reasons why they might like one ear over the other besides just random chance. The varieties could be very, very different. You know, imagine popcorn and sweet corn. Those are both corn, and yet they're very, very different tasting. And you wouldn't put sweet corn in your popcorn popper, and you wouldn't try to like bite into some popcorn on an ear and think it should taste like sweet corn. Sometimes in documentaries they make that mistake. So you have to make sure that the differences are minimized and just restricted to the things you're interested in. So what scientists do if they're trying to study a genetically engineered crop and make sure that the differences that they find are due to the trait that they're looking at, they have to use something called an isoline. That means that it's isogenic. It has the same genes. So what that means is that you've got two very, very genetically similar related varieties of corn that share a lot of the same genes and have a lot of the same characteristics. But the big difference between them is that one of them, and I won't say which one is which, one of them has the genetically engineered traits and the other one doesn't. So then rather than testing all those different characteristics like sweet corn versus popcorn versus field corn and all things in between, you're just testing that genetic difference you're interested in, and that is the GMO traits that have been added to one variety. Now, there are other kinds of differences that have to be controlled as well, such as where the plants are grown. We instructed Monsanto to grow them in plots that were near each other, not exactly side by side, near each other so that they were in the same growing environment. We also had them spray it with herbicides and pesticides as needed according to how a farmer would be growing them, and to treat them similarly, and to make sure that the one that was GMO was actually sprayed with Roundup, with glyphosate, and that the non-GMO one was not sprayed with that, which, of course, if they sprayed the non-GMO one with Roundup, then they wouldn't have any plants growing anymore. So what this does is this allows us for the when we're doing the experiment, if we find a difference, to say it is most likely due to these traits that have been added to one of them and not due to one of these other outside factors. So that's really important. There are a lot of studies out there that are published where they said, well, we took some soybeans and we got some other soybeans from somewhere else and we found a difference, and oh, it must be because of this one trait. But there could be hundreds to thousands of different kinds of genetic differences between those different soybeans. And without isolating those variables, without getting genetically similar isolines, growing them under similar conditions, they can't actually say that it's due to this difference that they're interested in. It's a really important thing. And it's a reason why we're doing this experiment and sending out the corn to everybody. I mean, we could always tell everybody, grab 2 ears of corn, but imagine if everybody got completely different ears of corn growing under different conditions. They don't know which one's GMO or not. That just wouldn't make for a good experiment. So we've done all the hard work of getting the corn grown in the right conditions and set up so that everybody can do the right experiment in their own backyards, and then we can collect all the data together. So got any other questions coming up yet? feel free to on the Google Plus page to submit some questions. Oh, okay. I got some actually coming through the YouTube page, actually.
Oh, I didn't even think to monitor that one.
Yeah. Well, I pulled it up just in case. So we got Mary. She says, "What has surprised you most about the response to this project at this point?" Actually, I was really surprised at how quickly we raised the minimum amount. It took just 17 hours. And that's counting the 8 hours or so that everybody was asleep after it launched at midnight. So, really, we're talking about more like 9 hours there.
Amazing.
How about you?
I think one thing that I've really appreciated, and I guess it doesn't surprise me because, if I may say so, Biology Fortified, readers and community members are, very science-minded. We do have a lot of scientists and science enthusiasts, so it's not too surprising, but I really appreciate some of the questions that we've gotten.
Yes.
On experiment.com and on Facebook and other venues, we've had people who have been really critical and asked questions. I love that people are bringing up the isoline question. Is often overlooked in studies about GMOs, and as plant geneticists, we know how important it is. So it's fantastic to see that people are, asking these questions. They're concerned about blinding, concerned about statistics, and things like that. And I just love seeing so many people involved in the making of this experiment.
Yeah. Just to add to that, I've seen and participated in a couple discussions where somebody said, "Hey, look at this project." Like, "Oh, well, this isn't going to work because, well, they don't have isolines." And then the person comments below that, "Actually, take a look at the project. They said they do." And then they're like, "Oh, righto, then this looks wonderful." Like some people have been rather English in there. "Oh yeah, so that's wonderful." Like, I fully support now. And it took a lot of thinking, and we had a lot of discussion about all these little details. And this is a big distributed experiment, and you have to find a balance between the scientific precision and the ability to actually have people do it in their backyards. But I really appreciate a lot of these critical comments. In fact, some people have micro-analyzed the video to point out to me that they could see differences between the ears, and to ask, "Hey, are you really using isolines there?" Which I'm like, wow. A fellow, a grad student who's just about to finish up in a few weeks from the same program, is actually in the same lab as mine. He commented on Facebook saying, well, like, you need to, I see some differences right there. And I see a difference in the cob color. If you look very closely in the very final shot, one of the ears that I use for the dramatization for the video has a different color in the cob. And that's because actually the two genetically identical ears I had that I was going to use, one of them got a little destroyed between shots, and I'm like, "Oh, I have to put in a substitute." So that is a dramatization, but they were microanalyzing it, and I really actually in a way appreciate that, because these are people who care about the science and having the science come out right. And so that's been actually really fun to see. Not necessarily surprising, but definitely something I really like to see.
Yeah, it's been great.
There's actually one other thing that surprised me, and that is one of our Twitter followers pointed this out to us, and we approached them. A Nobel laureate commented and said, "Hey, this looks like a really good project to do." And so then he sent an email to us, and it actually just got added as an endorsement on our project. And it's Barry Marshall, who co-discovered that Heliobacter pylori was involved in the formation of stomach ulcers. And he said he really likes how we turned around the Frankenfood slur and made a fun site about GM foods. He said the Corn Project might be a template for all kinds of experiments which kids can do to learn about science. As the data comes in, you should have a site for the analysis and include a statistical interface so the kids can run their own hypothesis testing, such as looking at results by postcode, effect of the label, and whether the corn on the left or the right was the best, such as, are squirrels right-handed? And I was just like, oh my gosh, we just got an email from somebody who won a Nobel Prize, like saying this looks pretty cool. Like, I was like, oh, that's cool.
Yeah, we'll have to look into whether or not that type of interface is available. But, you know, I have seen websites where people can enter variables and test hypotheses and run like a simple t-test or something. So maybe we can do that. That's a great idea. We'll look into it for sure.
Yeah, and I'd love to be able to have the funding to pay a programmer to do just that and have kids play around with data. I imagine, like, you know, your kids would just blow everybody away at the science fair if they made their own graphs of their own little hypotheses to, you know, to look at that. And maybe there will be a child who will discover that, you know, maybe the squirrels prefer corn on the right. And
So I think that, you know, talking about funding in the future and more experiments, I think that's a great segue into something that I wanted to talk about during this conversation. You know, we've talked about this being the first of many projects. You know, citizen science is near and dear to both of our hearts. You know, it's huge, people interested in get kits like this, in doing other experiments into classrooms all over the country, maybe even all over the continent. You know, that would be, I think, really beneficial for science in general. You know, that doesn't even have to be all about GMOs, just about agriculture and food. There's so many topics that we could explore. Yeah. But it really does all come down to funding. So we're definitely looking for grant opportunities, you know, anything like that. If people know of opportunities for us to apply to, or if they know of any foundations that might be interested in citizen science projects, you know, one thing that's really exciting is that now that we have one going, you know, we're developing our processes. The software that's being developed for this can be reused. We're making contacts with organizations like Statistics in the Community. And so all of these things will mean that the next experiment and the next and the next, but of course it all takes funding, and you know, our supporters have been so generous so far. Yes. The budget for this has been really tight, and really it's not paying for any salaries except for
just making labor to put together the kits.
Yeah. So pretty much all of this is volunteer for Karl, and Karl's done a huge amount of work. So thank you so much. You know, I'm sorry I'm not there to help lug 50-pound boxes of corn.
Yeah. Well, the crew is actually showing up tomorrow. Just finished hiring some help, and we're gonna have a small crew of people, and we're going to turn half of my house into a veritable factory. It's going to be Corn-mageddon over here as we put together 1,250 kits. But yeah, you know, the
photos. Who knows? Maybe it'll end up in a documentary someday.
Oh yeah, well, you know, we might post a little clip of the process to give people an idea. You know, this is what we're going through. This is how we're ensuring that we know where the corn is going. So yeah, well, yeah, this is going to be very visually oriented. Everything from the production of this to the data we get is all very visual. So yeah, I hope to be able to show a lot of that. But yeah, like, that's so great. You know, you said we want to do more of these. I'd love to do a big experiment like this every year. I'd love to do more experiments like this, perhaps on a smaller scale, and other times of the year. And we want to get ideas from the whole community of citizen scientists who are doing this with us for what to test next. Because in about a week or so, we're going to get these kits that we're going to start mailing them out, and it's just going to go in a big truckload to the post office, and it's all just going to get shipped out, and then it's going to get to people's houses in 2 to 3 days. Like, this experiment is about to happen. And then it's, you know, a few days later, most of you may actually be done with your experiments, and like, okay, now what do we do? What do we do next? And so we want to get pique people's curiosity and get more ideas of what experiments could we do, what sort of things could we study with lots of people, and let's try another one, and let's get the process rolling, and let's do another experiment, and then another, and then another. And And then somewhere along the lines, we'll find something that'll get us all some big prize. We'll have 1,000 people sharing a Nobel Prize or an Ig Nobel Prize, whichever one we get, right? So I think we have another question. Actually, this is good. I was talking about the kits. So Denny asks, do you guys know how many kits you'll be sending out yet? So we right now have about 350 people who've signed up already to get kits, and we have the funding to send them all to everybody. So yeah, we're ready to send that. When the fundraiser is over, we get the funds. We're going to pay for all the shipping costs, print out the labels, slap them on the boxes, and fly, my pretties, and they will go out to everybody. But then we're going to have another 800 to 900 more, depending upon how many ears we end up actually having in these boxes. We're going to have 800 to 900 more that we can still send out. So we'll still make it possible for people to sign up and to do the experiment this fall, and then we'll do some more in the spring. People want to be a part of that, and then we'll have so much data. It'll be fun.
Hey, you mentioned doing the experiment in the spring. So I did want to give a shout out to all of our friends in Canada. I know that we've had quite a few people who wanted to participate, and we just wanted to let you know we haven't forgotten about you. I've been investigating the Canadian phytosanitary requirements, and we understand that Canada is concerned about all plant materials entering their country, that we don't want to accidentally bring new pests, new plant diseases or insect pests into the country. So we're still investigating that, and we'll be looking into it. And we think that it'll be a few weeks before we get approval, or before we even let us do. So we know that it's getting cold up there quickly, and the animals are start hibernating and such. So hopefully by springtime we'll have that worked out. And if there's any Canadian officials listening, look upon our experiment favorably, and hopefully we'll be able to make that happen. We definitely want to, have our Canadian.
The ears are very clean, as you can see.
We have a phytosanitary certificate from the USDA when it came from Hawaii to the continental U.S. They are very clean. They don't harbor pests. So we'll do our best to make that happen. Yeah,
and the corn was grown in Hawaii because so Monsanto proposed that they could grow it in Hawaii because they could get it to us a little bit sooner. Because if they're going to grow it in the mainland, based upon the planting time, they would be harvesting it around now, or a little bit before. They would have just harvested, and they'd have to dry it, and it wouldn't be ready. But they were able to plant it earlier in Hawaii and get it out to us by October, so then we could actually do this experiment. And so we we're very pleased that that our readers and fans up in Canada want to be a part of this, and we want you to be able to be a part of this. So we're gonna get somebody who can be the point person up there in Canada, and we'll go through that process, and we'll get it to you for an experiment in the spring. We'll try to make that happen, but it's just the time it's going to take right now. There's going to be snow on the ground, maybe a foot deep before we can get it there, and that might not make for a very good experiment. I mean, the squirrels around here, they hop around, all winter time looking for food, so maybe they'll be extra hungry. I don't know, but it
might compromise the results. I mean, we are collecting, encouraging everyone to submit weather data along with their data, and I think that whole ton of deep winter weather would potentially screw the results. So we're going to be careful about that.
Yeah. I mean, it doesn't make for a good experiment if there's a foot of snow on top of these ears of corn.
Yeah.
But yeah, we're going to try for that. So we actually got another question here. From Jefferson again, a cynical comment. Anti-GMO activists will claim that a crowdsourced experiment is being astroturfed by Monsanto shills. Is there anything being done to defend the project against such claims? Yes, blinding helps, but of course, we shills will be given access to secret lab gear that lets us distinguish which sample is genetically engineered. Winky smiley face. So,
oh dear.
I thought I would just read that one in its entirety. So yeah, so that's a very good question. And we've certainly gotten some comments about that. Like, well, you guys are scientists and you don't say that GMOs are evil on your site, so you must be paid to say that by Monsanto or something like that. I mean, anybody who knows our site well knows that we've been independent from the very beginning. Now, that's a very good question, though, about how can you be sure that the blinded samples are really what they are. You know, let's just play devil's advocate or Monsanto's advocate or however you might want to do it. Let's just play this game for a second. So you get 2 ears of corn and you don't know which one's which. You've got a barcode. So you got a number, you submit the data, and then we hand it off to someone else and we tell you that they don't know which one is which. But then let's say you don't believe us. You know, or you think like, well, maybe they've somehow worked something out where they could, you know, they could switch the numbers in the data once they see what it is. It's something sinister and sneaky like that. So what's the solution to that? The solution is that we're actually going to tell everybody what the numbers are. We're going to provide the barcode information for every experiment kit before the experiment begins. But we're going to do that by uploading a password protected file. So anybody and everybody can download this file. And when the experiment is over, we will then give you the password. So you can see.
Key or whatever we work out.
What's that?
Or the encryption key.
Yeah, password encryption key, you know, so you'll be able to have the file and it'll have all this information. And then, of course, all of the data, the pictures, the results of the analysis and all that, all of this is going to be published. And anybody can go through and double check the results if they want to. In fact, I hope people do, because if we make a mistake, I would want to know. Because I didn't spend, a decade of my life going through, getting my bachelor's and getting my PhD and all that just to rig a corn experiment with squirrels in people's backyards. I mean, I'm in this because I want to know when I know something and know when I don't know something. And if you just go through all this trouble and then just switch the numbers around, then you're not doing science. And I'm here to do science. I'll let somebody else play around with the numbers. I mean, I expect there are going to be some people who are just going to say this, but that's not what we're here to do. That's not what we're here to do. And
I can add a little bit
transparent.
I've seen a couple of people comment on BioFortified blog that if the results show that there's no difference, then we might have a hard time finding a publisher. I don't know. We'll see. I think the citizen science aspect is unique enough to where some reasonably reputable publisher will be interested. But yeah, it's true. Negative results, as in, showing no difference, it's less exciting. And so in some ways, I'm kind of rooting for a difference. Who knows? Yeah. Maybe the squirrels and bunnies and whatnot will prefer non-GMO or GMO, and we'll see what happens. We'll just have to let the data fall out where it will.
Yeah, I mean, the funny thing is, if you think about, if you could desire one outcome over another, for a scientist doing an experiment, you want to find a difference. You want to find something new no one else did. If we were the ones who found that the squirrels wanted to eat one and not the other, and we showed it was really clear, well, then guess who gets all the NIH funding to figure out why that happened? And
but yeah.
Who gets the Nobel Prize in Physiology and Medicine for discovering this amazing squirrel aversion phenomenon? I mean, if anything, my personal interest is to make sure that happens, So we're that's why we wanted every transgenic trait we could find, make sure that the transgenic one, the GMO, is sprayed with Roundup and the other one's not, because some people say, well, maybe it was because Roundup was sprayed on it. That's the reason why. So we're trying to make it as likely as possible to find a difference if it's actually there. And if there is no such difference, great, then we can move on to the next question and we'll find something.
We'll need a follow-up experiment if there is a difference, because we have glyphosate and GMO in the same
and 8 different GMO traits.
All those different BTs and the different herbicide resistance traits and any marker genes. So if it does turn out there's a difference, we'd have to do a pretty complex randomized block experiment to actually be able to see what it does. But I think that we'll definitely be able to put to rest whether it's, GMO or non-GMO, and then we'll have to, do a more detailed experiment if there is a difference.
Yeah. So, the science is about like trying to find out what is true or not through gathering data and then making those conclusions based on that data. And whatever happens is reality, because that's what science is about. So I'm prepared to accept whatever result it is. And even if the result is the data is so messed up because we did this with people in their backyards that we just can't even make a conclusion, well, you know what? We still succeeded, because then we showed everybody how hard it is to do an experiment and the steps and the planning involved. And it's still an achievement as far as science outreach goes. And, just like you said with a journal, if a journal isn't interested in publishing negative results, there will, of course, be journals interested in publishing how we got negative results with 1,000 people in their backyards and with now I think about 50 school classes have signed up. But, by the time we send them all out, maybe it'll be 100, and how we involve so many people in this and they learn something about science. Somebody will publish that.
Yep, I think so.
Yeah. So let's see. Do we have any other questions coming in?
I think Rose is almost ready for me to come and put her to bed.
Yeah, and we're about at the 1 hour mark here. So far I don't see any other questions. So far, I think we're gonna I think we're gonna have a lot of fun. I mean, certainly over the next week at my house, there's going to be a lot of fun with all the mayhem that's going to happen here with assembling over 1,000 kits. I mean,
everything, Karl.
Yeah, I've been taking wheelbarrows full of these wooden blocks into around the side of the house after I've cut them and drilled them to bring them around to the basement so everybody can have the same setup. But yeah, there's oh gosh, there's so much stuff that we're going to be able to put up about this. Things we didn't even mention. So I mentioned we know what the crops have been sprayed with. So we're going to publish what the spray data is, so people know exactly what conditions and what days and what the weather was like, how the crops were grown. There's a map of where the strips were grown. There are loose kernels in some of the bags that these come in, and we're going to take those loose kernels and we're going to be able to verify that there wasn't significant cross-pollination between them to make sure that the GMO is the GMO and the non-GMO is the non-GMO. And so there's all kinds of things that we're going to be able to tell people about and show people how we're doing this. I'm actually really excited to be able to tell them about all this. And we're going to publish the MTA. We're going to publish all of these things. It's going to be really fun. So Denny asks another question. Will you be doing more Q&As once the data gathering gets going? Smiley face.
I think so.
Yeah, I mean, I think that would be a good idea. So we're going to try not to release the full results until we've given everybody a chance to send their data. But we're thinking about ways that we can show people some of the process, even if it's to set up a one experiment kit on a webcam and let everybody watch it like live on YouTube and just that'll be a couple of live ones or something in my backyard, let's say, and then we'll let everybody know eventually which one was which or something. We're thinking.
I want to see what your turkeys think, Karl.
What's that?
I want to see what your turkeys think.
Oh yeah. I haven't been able to convince the turkeys to get corn off of whole ears yet. Yeah, but Anastasia points out that I've got some turkeys that live in my neighborhood that come around. So not only do I have squirrels and chipmunks, but a family of turkeys, and they've managed to find a few handfuls of birdseed and such for them to eat. And I've got to see them grow up from little chicks all the way to adults where I can't even tell them apart. And so, so who knows? Maybe we'll get some footage of turkeys pecking at some of these ears of corn. Who knows? Yeah. But yeah, more Q&As would be good. We're going to give people very clear directions on how to do the experiment. So make sure that everybody does it well. And so yeah, there will be some more room for interactions about this. This won't be the last.
Yeah, you're going to post a video about how to put the kits together, right?
Oh yeah. I'll have a short video on how to do that, and also tips on where to put the feeding stand, and some do's and don'ts, such as like if you put it on a fence line and you just put it like right on the fence like that, the squirrels may approach from one side and eat that ear first. So things like putting it on the fence like this and putting a nail through it to hold it there. So when they go up to it, they see two ears and they have to choose left or right, GMO or not. Little things like that to hopefully make everybody's data as good as can be.
Awesome.
Yeah. So I think that's pretty good, unless you got any other comments you want to make, Anastasia.
No, I think this has been a fantastic conversation. It was nice talking with you, and I'm really looking forward to seeing the data start coming in. Oh
yeah, so am I. The thing I can't wait for are the thousands of suggestions for what we do next after this.
Oh yeah.
Because then that's just going to be a whole world of possibilities of other experiments we can do with all of you. And we'll design those together.
Yeah. All together. Right. Citizen science.
Yeah, citizen science for the win. We'll see. We'll see as the data comes in, and we'll see whether or not we got in over our heads and started something that it could be a corn train wreck, a corn apocalypse.
Oh dear.
I got all kinds of corny names to use because I've got 2,000 pounds of corn over in the next room in boxes. I've been thinking a lot about just how much there is here. Yeah.
Okay, great. Well, thank you very much, Karl. Have a wonderful night, and have a wonderful night, all of you listeners. Thank you for tuning in.
Yes. Thank you, Anastasia, for being part of this. Thank you, everybody, for your support. Please tell people about our experiment and sign up if you haven't already. In particular, tell some teachers, because we're going to send the kits to them for free, and they can do this experiment with their students, and everybody will learn something one way or another. All right. Thanks, everybody, and goodbye.
Bye.